So our next speaker for this morning is Dr. Jennifer Below. Dr. Below is an assistant professor in the Vanderbilt Genetics Institute. She is responsible for the development of new tools in genetics data analysis, including IMUS, Primus, and Padre. Dr. Below focuses on leveraging high-dimensional omics data, deep phenotyping, and EMR-based DNA databanks to uncover new causal risk factors for complex diseases. She is a leader in exploratory research into the genetic risk factors for apical periodontitis. Please join me in welcoming Dr. Below as she presents Leveraging Big Data to Fuel Precision Health Research. Dr. Below.
Hi everyone, I'm getting over a cold, so forgive me. Talking for an hour is going to really sort of max me out, so I'm going to be sipping on this coffee during the talk. See if I can keep my throat clear. So today I wanted to talk to you guys. We've seen a lot of slides talking about all of these sort of inputs into precision health initiatives in public health and especially in dental oral health. And today I'm coming to you not as somebody with much experience at all in dentistry or oral health, but really somebody who has been working and devoting her career to genomic medicine and the genetics bubble in those arrow diagrams that all go into that big data precision health. So I wanted to tell you a little bit about where we are, what tools we have at our disposal, how we're leveraging those tools to improve health disparity research, and specifically some of the resources that are available publicly, even to you today. I went ahead and dug into a couple of our large databases that are publicly available last night, and I just spent about 4 or 5 hours tinkering around and pulled out a bunch of genes that are potentially very interesting for some of the traits that you guys might be interested in based on the conversations I've seen over the last couple of days. So truly cutting edge, like literally last night at midnight, genetic medicine. So I wanted to kind of review some of the primary concepts within genetic epidemiology. We had a phenomenal primer yesterday that took us all the way down to sort of genes and chromosomal architecture and how genes are structured, AGCs and Ts, and we also had some pretty amazing high-level talks about pediatric genome-wide association studies and some preliminary data that were coming out of those. But I wanted to kind of bridge the gap. How do we get from the AGCs and Ts to a genome-wide association signal that we believe in? And is genome-wide association really the only approach that we have there? So I also wanted to address a little bit about family-based genomic studies, take you through the story of some new approaches that are really taking the genetic architecture and reorienting them around gene function instead of single nucleotide variants, and then how we're currently today leveraging biobanks and EHR repositories for these phenome-wide characterizations. I think you guys have seen the word phenome-wide or PheWAS on a number of slides. I wanted to kind of take us through that a little bit more in a little bit more detail. So the idea of genetic epidemiology is that we're really expanding the parameters of classical epidemiology, where we want to understand the patterns of disease occurrence in human populations and the factors that influence these patterns. So classical epidemiology triad is going to have your host, your agent, and your environment, who comprise the who, what, and where. And in typical epidemiologic research, it applies well to infectious disease. However, for genetic epidemiology, the agent, host, and environment are all slightly shifted here. The environment becomes these extrinsic factors, and we also have the host's intrinsic factors that are important to characterize. So the host becomes really a conglomeration of all of these susceptibility genes or potentially protective genes, and we have the physical conditions, and we have the lifestyle choices that the individual makes. We want to put these together to understand how disease emerges. So for me, genetic variation is the exposure of interest. It's intrinsic, it's there from birth. Sometimes it's causal, but sometimes it's not necessary or sufficient for disease risk. And this term of genetic epidemiology is truly an umbrella term. It doesn't just refer to the genetics, it also refers to all of those environmental components and their interactions with the genetic components, and the gene-by-gene interactions that are also happening. I thought about making a slide for this, but instead I'll just use my arms. You can basically just say you've got health on one side and disease on the other side, and you're going to have this pileup of factors, environmental and genetic, that are going to push you down towards being healthy or being at risk genetically. Sometimes, like in the case of a Mendelian disease, the genetics are going to overwhelm everything, and it doesn't matter what your environment is, you're going to end up being sick because it's high penetrance, allele that completely obliterates gene function. On the other side of the spectrum, you have things that are more complex, like type 2 diabetes or metabolic disease, obesity, where an environment can completely overwhelm the genetic risk factors, and you can end up healthy even when you have susceptibility inborn. It's important to recognize that the genes and the exposures can happen across many time points at life. You know, you can have exposures in utero, you can have exposures that come later in life, and exposures can, the genes can interact with disease severity, genes can interact with disease both in terms of your susceptibility to the disease, the onset of the disease, the severity of the disease, and all of these genetic interactions influence actual health outcomes. So in the spectrum of genetic epidemiology research, you have this sort of false dichotomy. On the one side, you've got these single gene disorders or Mendelian diseases. These tend to be very rare. They're often caused by a single mutation in a single gene with really large effects. On the other end of the spectrum, you have complex diseases, which are generally speaking pretty common in the general population. They result from multiple gene interactions with environment that are all relatively modest effects that are harder to statistically identify. And we can see sometimes non-additive effects between genes and environment. You can have multiplicative effects, and these two get more complicated to model, statistically speaking. So our objective in genetic epidemiology is to really understand the whole picture of disease etiology. But specifically, we want to make sure that we're including and appropriately accounting for the genetic markers to directly measure risk and characterize the underlying sort of macromolecular effects that are happening that are increasing risk on a biological pathway. The steps for performing genetic epidemiology research is to, first of all, we have to establish that there is a genetic component to the disease. Is this going to be a phenotype like accidentally getting hit by a bus, where you might argue that it's, you know, purely dumb luck or environmental conditions that resulted in the health outcome. But I remember when I was a graduate student and my professor asked me, can you name a way to die that is completely environmental and has nothing to do with genetics? And I said, getting hit by a bus. And she said, well, if you're getting hit by a bus, you're probably jaywalking. And if you're jaywalking, then you probably have risk-seeking behaviors, and that's genetic. So in my world, everything's genetic. So first we want to establish that there's evidence of a genetic component to the disorder. We want to actually measure the relative size of that genetic risk in relation to the other sources of variation in disease risk, which allows us to design a study that's going to be well-powered to detect those specific genetic risks of interest. And then we want to be able to actually identify not just a particular variant, but the gene itself, the functional unit of the genome that is responsible for the genetic component of risk. So how do we go about establishing that there's genetic components to the disease? You know, if you're looking at something like tooth caries and you see that these so strongly influenced by socioeconomic factors, obesity phenotypes, and you know, the microbiome of the mouth, you might be tempted to make the argument that this is not a particularly genetic trait. This is going to be more environmental. And the way we measure how genetic a trait is by looking for familial clustering. But familial clustering can still be due to either both the shared genetic environment, or it could be due to the actual non-genetic environment. We measure the size of the genetic effect. We look for evidence of a particular genetic model, whether it looks dominant, recessive, or polygenic or complex. And then we work on finding the disease gene. And once we find the disease gene, there are many sites in genes that are not particularly influential of outcomes. Coding regions tend to be more influential. Promoters, stop sites, splice sites, going back to the talk that we heard yesterday, looking at the actual architecture of the gene, but many variants within genes are completely tolerated and will have no effect on disease risk. And then finally, we want to understand how does the variant or the variation within the gene actually contribute to the disease in the general population. It's one thing to say, I found this one family from a rural village in the Middle East, and they seem to have a tooth agenesis phenotype of interest, but if the genetic variation within that family is not necessarily seen within general populations, the impact of that discovery might be isolated to just that single family. Uh-oh. Either my, oh, there we go. So we can sort of, like, in the same way that we artificially divided traits into rare traits and common complex traits, we have different statistical strategies for uncovering the underlying genetic susceptibility factors for those two sort of different ends of the spectrum. On the one hand, if you've got a variant of very large effect, high penetrance following a traditional genomic model like recessive or dominant, we're well powered in the statistically when we perform linkage studies, which looks for co-transmission of genomic regions with the disease inheritance within a family. And on the other end of the spectrum, you have what we've been hearing a lot about at this conference, which is association studies or genome-wide association studies. And in genome-wide association studies, what we're doing is we're searching for genetic differences between population members who have the disease cases and those who do not, or controls. So I'm a huge proponent of family-based genomic analysis because it's so tremendously powerful. If you look at complex disease genetics and you look at the literature, just a few years ago, a paper came out on type 2 diabetes where they looked at something like 280,000 people, and they looked at the genomic contributions of type 2 diabetes, and I think they might have found 7 new variants. 280,000 people. 7 new variants for type 2 diabetes. The risk, not the risk reward, but the cost versus reward balance there feels a little bit off to me. For perspective, in a family-based genomic analysis, if the variant is of large effect, you can discover a new gene for a disease with only 10 people. 10 versus 280,000. So these kinds of studies can be extraordinarily informative for understanding the genetic architecture of disease risk. And just because a variant in a gene might be private to that particular family, you're still learning incredible information about the pathways and the architecture of risk that occur naturally without it necessarily being a particular variant that's seen elsewhere. So in order to perform familial analysis, first you have to establish that the disease or trait does cluster within families. And in order to do this, we can compare the disease frequency in relatives of affected individuals with the general population, or with disease frequency in relatives of unaffected individuals. And so my lab has collaborated with the University of Texas Health Science Center at Houston and the School of Public Health to perform traditional, old-school, classical genetics on tooth agenesis phenotypes. We have a couple of families from the United States and about 30 or 40 families from rural areas within Turkey. And all of these families are extremely multiplex. Here in the pedigree, you can see all of the sort of 3/4 filled in individuals represent cases that developed, that failed to develop 3 or more teeth. And doing this, we were able to identify a number of genes for familial tooth agenesis. In this case, the gene ended up being a known gene called PAX6, but we also identified evidence that was suggestive of some new genes that are involved in this pathology. And again, to kind of merge this in with where we are in terms of the big data, small data analysis, this gene was found. The people that are colored in green here are the only people that we genotyped. And the two individuals that are in yellow, you can't probably, yellow's a terrible color for slides, I apologize. But those two individuals there were the only two that we performed sequencing on, whole exome sequencing. So the entire cost of this project was probably around $4,000. And we only generated genetic data on a handful of individuals, and yet we were able to find not only the causal gene, but the causal mutation that led to the phenotype. So in terms of cost-benefit analysis, if I could find families like this, I would study them every day of the week. Forget about these GWAS projects. I love family-based genomic medicine. So this is just a rundown from the 15 families that we published most recently. All of the genes that we ended up identifying causal variants in, and what those variants were, and whether or not the zygosity was heterozygous or homozygous for the causal variant in the family. So we see that this phenotype is both driven under recessive models as well as dominant models. So this is just to dip your toe in the pond of, please don't forget about familial genetics. On the other hand, you've got more complex genetic analysis. You can have familial aggregation where there's not actually a large, highly penetrant genetic susceptibility factor co-segregating through the pedigree with disease risk. And you can also have some really interesting cases of gene-environment interaction. For example, my favorite story of this from my personal experience was my lab was involved in the discovery of the very first gene for mesothelioma risk. Mesothelioma is the cancer that is asbestos-associated. And what we found was that in these families, there was 100% penetrance, which means that every single person in the family that carried the susceptibility variant for mesothelioma went on to develop mesothelioma. And so we were wondering, have we found a genomic variant that has action in the absence of the known environmental susceptibility factor, right? We had no knowledge that these people had pervasive exposure to asbestos. They were not asbestos miners, they were not pipe fitters. But in mouse models, we found that if the mouse had the genomic variant that we identified, it was in the BAP1 gene, and we kept the mouse in a completely sterile environment, they would never go on to develop a cancer. And if the mouse had any exposure to mineral fiber, needle-like mineral fibers at all, they would go on to develop mesothelioma. So it just turned out that the threshold of exposure was so low that every person in this room probably has at least that exposure level to mineral fibers that would have been sufficient to kick off this gene-environment interaction. So the environment can be a pretty low bar, and that's an important thing to keep in mind. So in the, in the interaction between exposure and response risk, basically this is a model to show you that if you have the susceptibility gene, but you have low exposure, your risk still remains low. But if you have the susceptibility genetic factors, and your exposure is high, then you're at much greater risk than either the general population or those that have the protective genetic factor. And this is sort of the classic model of how gene by environment interactions work. So finally, you can have other evidence that phenotypes aggregate within families other than just an enrichment. An earlier age of onset is frequently a hallmark of a larger genetic factor contributing to risk within a family. And you also have stronger phenotypic correlations between parents and biologic versus adopted children. So these are all sort of things to keep in mind as you're looking at data within clinics or within public health data repositories when you're trying to think about what the best strategy is for uncovering the genetic susceptibility factors at play. So we've kind of gone over most of this, but the next sort of logical step here is to be moving away from family-based designs into population-based designs. And here you can see that as we've had extraordinary data growth within human genetics as a field, and this is fueled by what a lot of people were talking about yesterday, the reduction in costs for a lot of the technology that allows us to do genetic analysis, people have sort of left linkage analysis and family-based designs by the wayside and have moved towards genome-wide association studies and whole exome and whole genome sequencing. So what really underlies a genetic association study design? If we have affected and unaffected individuals, and the goal is to identify whether or not a particular SNP allele occurs more often among cases than compared to controls, what we need to do is we need to measure the genotype in a bunch of sick people and in a bunch of healthy people and look to see if there's a difference in allele frequencies between the sick people and the healthy people that is suggestive that, that the variant is either itself or something nearby it that is in linkage to disequilibrium with it, contributing to risk to the disease. The advantage of this approach is that it's relatively easy to collect data from individuals rather than whole families. As the prices of genotyping have dropped, the prices of studying families has stayed stable. It is still just as expensive to have to track down these multiple individuals that are all related to one another. My lab has addressed this issue by developing software tools, which got mentioned in my very kind introduction, Primus, pedigree reconstruction and identification of maximum unrelated sets, and Padre, which is a distant relationship estimation algorithm, so that you could actually, without any clinical knowledge of the particular relationships between individuals, for example, in a large DNA data bank or a large repository, be able to identify those relationships using the genetics, reconstruct the pedigrees, and then potentially move on to do family-based genomic analysis. But in general, ascertaining samples for a project from a hospital or other general population-based approach is much cheaper than going to the work of collecting family members. A disadvantage of genetic association study is that your power really depends on a combination of factors: the effect size of the variant on risk of disease, the allele frequency, and the sample size. So if your effect size is relatively low, you have to have a very common variant and you have to have a lot of people in your study in order to be able to detect it. If your effect size is relatively high, then you can get away with detecting rarer variants in smaller sample sizes. So there's sort of a proportional relationship there that results in the power that you have to detect. And so as a result, in order to detect more and more variants, people have been moving towards larger and larger studies. Another disadvantage of genome-wide association studies is that they're oftentimes really hard to interpret biologically, and I'm going to go into this later in this talk. And then an issue that was brought up the other day was the issue of population substructure, admixture, and the biases that that can introduce. And so I wanted to devote a little bit of time this morning to addressing that sort of head-on. So I know that this is not an oral health or dental phenotype, but I wanted to take a few minutes to really kind of delve into a couple of issues that were brought up that got me kind of worked up yesterday. So basically, a lot of people had brought up the issue that precision medicine has left minority groups behind and ethnic minorities behind. And also that admixture can introduce biases that can invalidate the results of genome-wide association studies. It's true that the number of minority patients that have been enrolled in studies of genomic risk has been orders of magnitude smaller than those that have been brought in that are Caucasian. But at the same time, I wanted to point out and highlight how powerful the genotypes are that do come from non-Caucasian populations, and really emphasize how incredible it is to be able to leverage that data in the context of disease gene mapping. And I also wanted to sort of address head-on that if you mishandle admixture in genomic analysis, yes, it can absolutely bias and invalidate your results. However, if you leverage it correctly, it's one of the most powerful tools we have. So I wanted to tell you this little story about what my lab is doing with type 2 diabetes research in multiple ancestry groups, and specifically my lab is responsible for the Hispanic analysis. And so we're part of this study called Diamante, which stands for Diabetes Meta-Analysis of Trans-Ethnic Association Studies. So PIs from across the globe have taken responsibility for each of these different ancestry groups: African, East Asian, European, Hispanic, Latino, and South Asian. My lab is responsible for the Hispanic Latino effort. And jointly, we're performing a transethnic meta-analysis of all of the data together. The aims of this are not only to discover novel variants and loci that are specific to understudied populations, but to increase our power to detect novel loci for type 2 diabetes across populations, and then also leverage the finer haplotypic structure because of the different recombination patterns that we have within the different ancestry groups to improve the fine mapping resolution. So if you perform your analysis just in Caucasians, which have a relatively coarse haplotypic structure, you're going to find a GWAS signal that could span megabases and potentially tens or dozens of genes. And you don't know which gene in that region is necessarily underlying your actual genetic risk. But by taking advantage of the differences in the linkage disequilibrium patterns, or the finer haplotypic structure, we're able, of these other ethnicity groups, we're actually able to fine map, hone in, and exclude many of the genes in those regions to be able to localize the gene that we think is actually functionally underlying that risk factor. So, as I mentioned, my group was in charge of the Hispanic Latino group. For us, this was exciting because it presented a unique opportunity to work towards correcting a major health disparity while improving our understanding of the genetics of these traits. And because I'm all about killing more than one bird with a stone, we reached out, and since we were contacting all these groups anyway, not only did we do type 2 diabetes as a phenotype, but we also did other cardiometabolic traits, including serum lipid levels, because we had the opportunity. Why not aggregate all of that data together? So this is a sort of small text slide, but this shows all of the different studies that we contacted and the samples that we aggregated across all of these different cohorts. We ended up with over 13,000 type 2 diabetes cases and 21,500 controls for a total of almost 35,000 individuals for this study. So 35,000 Hispanics in a study is certainly nothing to sneeze at, and upwards of 30,000 individuals each for the lipids traits. Yet what we found was a lot of replication that we had seen before from the Caucasian study. So you might, at first glance, think to yourself, well, that's not particularly exciting. You found a bunch of known stuff. But we found this stuff for the first time in Hispanics. They had never been, these genes and these loci had never been identified in Hispanics before. And so this was the first evidence that there's not necessarily as much disparate architecture of these traits as you might think at first glance. That indeed, the same physical spaces in the genome that are conferring risk in Caucasians are also conferring risk in Hispanics, and I believe that this is going to be true across the complex disease trait landscape. But what's interesting, particularly, is that at many of these sites, it is not the same genetic variation as in Caucasians. So the physical location, that gene, that LDLR, LDL receptor gene, is still the locus that is conferring risk of elevated triglycerides, but the haplotype, the actual segment of DNA that underlies that signal, is unique. And I think that's fascinating. It's something that my lab's going to be working really hard on following up over the next couple of months. But the most interesting story out of all of this is actually going to come from this transethnic meta-analysis, where we're leveraging all of these different populations. So you can see here how many different studies each different ancestry group was able to pull together for this enormous transethnic analysis. And the transethnic analysis, I just told you, I was so impressed that I was able to get 35,000 Hispanics. But look at these numbers for the total sample size, right? We're up to about 1,250,000 people now in this study of type 2 diabetes. So I like to joke that this is kind of big data analysis the hard way, where we had to collectively reach out to 120 different groups and convince them each to contribute their genomic data to this effort. So you might also call this herding cats. An incredibly challenging way to go about big data analysis. And I think there are probably better ways and more efficient ways than this, but this will certainly become one of those papers with 8,000 authors on it. So this is just to give you a sense of the diversity within the different populations. And I think that it's worth noting that the African Americans, Caucasians, East Asians, South Asians, and Hispanics, even within the population group of Hispanics, we see an enormous amount of diversity within the Hispanic population. So this is a principal components analysis. We're basically looking at the variance between, if you aggregate all the genetic material that we had access to and you analyze the variance in that genetic material, these kinds of plots illustrate that graphically. And so when you see tight clustering, the allele frequencies within this population set of studies is very tight, but you can see that with this enormous spread that Hispanic populations are actually extremely heterogeneous. And that's also both something that makes us more powerful for this fine mapping effort, but it's also something that means that we have to be more careful with our statistics so that we don't misuse that admixture and introduce bias. With 1.25 million individuals, we were able to recapitulate pretty much every single known loci, previously described loci for type 2 diabetes risk, and we were also able to identify 40 novel loci. In blue, purple, whatever, you can see the novel loci, and in gray, you can see the known loci. And we were able to then do some dissection of the distinct association signals at these susceptibility loci. What we did was we did an iterative approximate conditional analysis. So basically we said, here's the SNP with the greatest GWAS signal. So this is the SNP with the greatest difference in allele frequency between the cases and the controls in this region. Now I'm going to include that SNP in my model, and I'm going to see if that SNP's variation explains the entire signal at that point in the genome, or if even after accounting for that genetic variation, is there still evidence of an effect coming from that part of the genome. And what we found was that at our 208 distinct loci in the genome, there were actually 342 distinct association signals, which tell us that these peaks are not necessarily driven by one genetic susceptibility factor, but in many cases, they're driven by many genetic susceptibility factors. This gives you a sense. 148 of our loci were driven by one signal, but 5 of our loci were driven by 5 or more signals. And to give you a little sense of this, here's the INS KCNQ1 loci. And after we've performed iterative conditional analysis, this is how many variants are actually contributing to the signal in this locus. So it's 11 signals coming from the KCNQ1 gene and 5 signals coming from the INS insulin gene. And again, this is all being powered because of the diversity in the sample. We wanted to do a heterogeneity analysis. So this is something else that's very well powered in environments where we have genetic heterogeneity and we have these different populations. We can look to see is the evidence from the genetic effects similar across all these different ancestry groups, or are some ancestry groups driving a signal where there's no evidence coming from other ancestry groups. We can sort of partition out those effects. What we found was that in general, in those 348 or whatever it was loci that I mentioned, most of them, the vast majority, have very consistent effects coming from all of the different population groups. But 55 of our association signals did show significant departures where they were driven by one or a subset of these ancestry groups. So this is 2 examples. Here you've got a variant within the LEP gene, and you can see that it's completely being driven by effects coming from the East ancestry group, and there's really no evidence of effect coming from any of the other groups. Down here is your odds ratio. And over here, you can see that all of the groups at this UBE2E2 gene show modest evidence of association except Hispanics, where we don't see any evidence of association. So these are some of the kinds of analyses that we can only do when we start looking across populations. So finally, I had mentioned that we can do fine mapping. So we can take these large linkage disequilibrium chunks of the genome where we don't really know what about that section of the genome is actually influencing disease risk, and we can narrow it down if we've got data sets where there's more recombination breakpoints that have broken up the haplotype and help us localize the actual functional variant. So we do this by defining 99% credible sets. And what we found was that at 139 of our loci, the 99% credible set in this analysis included 10 or fewer variants, which is an extreme reduction in the number of variants at each of these loci from what we see when we're only looking at Caucasians. And for 36 of these loci, the 99% credible set, so the variant that explained 99% of the variants in the locus, was driven by only one variant. So in these areas, we're able to completely ascribe the signal to a single variant that's causal for the signal. We did some analysis of what these variants that are driving these credible sets are doing in the genome to look for function, and what we found was that there was enrichment, that these particular markers were falling within coding exons, which makes some sense because you'd expect that markers that fall within coding exons are actually going to have a larger effect on phenotype. And we also found evidence for particular enrichment of transcription factor binding sites for these two specific genes. And to walk you through a little bit about what this sort of fine mapping effort really looks like, so here's an example of a region in this DGKB gene. And here's our variant that has the highest evidence of association. But this red indicates that these other markers down here are in very strong linkage disequilibrium with this variant that's shown in blue. So we really can't tell across this region what is the variant that is explaining the risk of disease. When we look in just Europeans, this variant explains 52% of the evidence of association in the region. But when you look at the transethnic data, suddenly this variant jumps up and explains 86% of the variation. And that's due to the finer haplotypic structure in the African Americans and the Hispanics specifically. And in fact, when we look to see, well, what does this variant actually do? It maps to a PDX1 binding site, and it's an EQTL for DGKB in human isolate cells. So this is very convincing evidence that this is actually the functional variant, and that we've narrowed this down from a region of the genome that is associated with diabetes risk to the variant that is associated with diabetes risk. So this is extremely exciting for us human geneticists to be able to hone in on the actual functional action that's contributing to risk. This is just a brief summary slide of this work, but the key point that I want to make here is that all of this was only possible because we brought together data from more than 1.25 million people. And like I said, we really did this kind of the hard way. And it's worth asking the question, what is the path to getting to the point where we can do this kind of analysis, but for other phenotypes? Whether your interest is, dental caries or gingivitis or other characteristics of oral health, how do we get to a point? Because I would argue that we're going to see trends of complexity equivalent to metabolic disease. The environmental impact is going to be as strong in many of the characteristics of oral health that we care about as we have in metabolic diseases. And if this is what it takes, how do we get there for dental and oral health? I think the answer is going to be that we need to start bringing together the data that exists and pooling data resources that emerge in the future. So like I said, we had to reach out to 120 different independent submitting investigators in order to get together a GWAS data repository, which would then allow us to do this large-scale meta-analysis of all of these individuals. But another alternative strategy, rather than having each data set emerge in its own silo and then having to go back and bring them all together, would be to start to leverage EHR and DNA databank repository type environments. But it's important to note that what we can do uniquely well in biobank data is slightly different than what we can do in these sort of independently ascertained, directed, phenotypically driven single studies. But the strengths of biobanks certainly are extremely powerful for understanding mechanisms of disease. So I want to go into a little bit of the differences and a little bit of the advantages of biobank research. So in a biobank, we've got large numbers of people We have an electronic health record for all of the people in that data set. And we've got opportunities to generate omic data, genomic variation data, transcriptomes, metabolomes, methylomes. And the question is, it might be too expensive, the burden might be too high to be able to generate all of this omics data, but it's possible that we could measure genomic variation and then impute from the genomic variation, we can estimate other characteristics of the omic data from that, which will help us functionally orient these analyses. So at Vanderbilt, we have an electronic health record called BioView, well, and DNA databank called BioView. Our electronic health record is stored in something called the synthetic derivative. It has approximately 2.7 million people in it today. And we have biological materials that are approved for genomic research in about 250,000 of those samples. Today, we have genotyped about 120,000 of the individuals with the biological samples using whole blood, and have been beginning to explore ideas of imputing transcriptomics from the genomic variation. So like I said, this kind of single phenotype genome-wide association analysis, if you haven't seen these before, this is a Manhattan plot where you've got the chromosomal location of the evidence across the x-axis and the log 10 of the p-value, and you see these spikes and these spires wherever there's significant evidence for genomic effects coming up. And this kind of analysis is great in cohort or cross-sectional study design, but that's not where our unique advantage lies in large-scale EHR databases. What we do really well in EHRs is phenome-wide association studies, or PheWAS, where we have a set of variants or a set of genes of interest, and we want to look at their effects across the entire spectrum of health outcomes stored in the EHR. So in a GWAS, we're really asking the question, what variants are associated with this phenotype? In a PheWAS, we're asking the question, what phenome is associated with this variant? I've already touched on a few of the issues that emerge when you try to do genome-wide association studies. But to reiterate, common variants tend to be uncoupled from gene activity. You can have a single nucleotide variant that's strongly and reproducibly associated with your phenotype of interest, but it exists completely in the absence of any kind of functional annotation in the genome. It's not a coding variant, it's not near a transcription factor binding site, it's just a variant in the middle of a gene desert, and you just have no idea what its functional impact is or what its mechanism of action is. And this creates an interpretation challenge for us. When we're testing all the variants in the genome, we're performing millions and millions and millions of tests, and we have the obligation of statistically correcting for those tests under a frequentist model. And that burden of test correction is high, which reduces our power. And the effect sizes per individual SNP are so small that we have to have huge data sets, which I've already talked about, and association signals can span multiple genes or no genes. So like I said, gene deserts, or it might be a gene-rich area, and we just don't know which gene it is. And the Hispanic analysis I showed is one way to get it uncoupling some of those problems. But another way is to start rethinking how we're performing these genome-wide association studies in the first place. So moving beyond single nucleotide variants and moving over to transcriptomics or gene expression, we can start to think about the factors that are influencing gene expression levels. Certainly, it's not purely genetic. For most genes in most tissues, there's usually an environmental component to gene expression. There's a component of gene expression that could be related to your age. Are you a newborn baby? Are you 106 years old? But there's some component of gene expression for every gene in every tissue that is going to be genetically regulated. For some genes, that portion of genetically regulated expression is going to be small, so the genetics will explain a small portion of that variance. For other genes, the portion of genetically regulated control, or the amount of variance that's explained by genetics, is going to be large. And the idea here is, can we estimate this genetically regulated portion of gene expression from the actual genotypes themselves? So if this really sophisticated drawing of mine represents a gene, and here you've got exons, and here you've got locations of markers that you've measured the genetic material, so these are SNPs. And we've also measured the abundance of a gene product, so the RNA level, in a specific tissue. We can do an association test to identify which of those SNPs are predictive of expression in that given tissue. And then what we can do is create an additive model of gene expression where we take the genotypes from each of these red markers and we fold them all together to create an estimate of the genetically regulated portion of gene expression. So when you see this GREX, that's what I'm referring to, genetically regulated expression. This was done, this slide's a little bit old, instead of in 41 tissues, now it's 52 tissues that were all brought in from this large-scale genotype tissue expression project called GTEx. So now what this means is that in a data set where I have not measured RNA product at all, I've done no transcriptomics, I've only done genomics, for my set of individuals, I can look at the genotypes at these different markers, and I can actually create an estimate of the relative expression of that gene. And as a reminder, for some genes in some tissues, this is going to explain a lot of the actual observable variance in the RNA abundance, and for other genes in other tissues, this is going to explain a relatively small amount of the variance in actual measurable RNA expression. But in either case, whether I'm explaining a lot of the measurable variance or a little of the measurable variance, I can do an association test between my predicted expression level and my phenotype of interest, whether it's a binary trait like tooth caries or a quantitative trait like pocket depth. Oops. So the advantage of this is that it reorients our genome-wide association results into directly interpretable, functionally oriented results. It reduces our multiple test burden from millions of SNPs to now just thousands of genes, and it allows us to aggregate those small effects that are coming from each of those different functional variants and collapse them down into a gene-based effect, which aggregates that evidence and makes it more easy to detect. This approach was published, it's called PredictScan, in Nature Genetics in 2015, and the reference panel that we've been primarily using for the expression data when we built these models all came from GTEx. Also, all of the code and all of the models built for all 52 different tissues within GTEx are publicly available on GitHub from Hockey's lab. So I've kind of walked you through this step and this step, but now what is so exciting is that we're actually able to apply these models in genomically characterized, but not transcriptomically characterized data sets, such as large EHR repositories and DNA databanks. So at Vanderbilt, we have estimated for every person in BioView, so for all 120,000 people that have been genetically analyzed in BioView, we have estimated their tissue-specific gene-level expression in all 52 tissues, and we are now able to compare that predicted expression level to their EHR record phenome-wide, so throwing that against every single trait, and create a results portal that allows us to query the results of the genetic effects by either gene or by phenotype. So the result here is that we're able to say, what does this gene do? What does the natural variation in the expression of this gene actually associate to across the entire medical phenome? So to walk you through, this is what I started doing at like 9 p.m. last night and finished doing it like 2 a.m. this morning, was to start to play with querying the transcriptome by gene or by phenotype. So just as sort of an illustrative gene, not necessarily one that's been strongly associated in dental phenotypes, but one that's sufficiently famous, the CFTR gene, which is associated or causal for cystic fibrosis. And you can see that when we drop this into BioView and we look for all of the health outcomes that are significantly predicted by the genetically regulated portion of expression at CFTR, you can see that these traits come up: cystic fibrosis, pneumonia, bronchitis, etc. So the phenotypic outcomes that result from dysregulation at that gene are exactly what you would expect from a gene that is going to have this lung phenotype. We can also query by phenotype, and this is where I thought it could get a little bit fun. So I went ahead and dropped into our BioView portal data repository the PheWAS code for gingival and periodontal diseases. And interestingly, this gene CTNS came up again and again and again across pretty much every tissue in GTEx as being significantly associated with p-values that pass our threshold for multiple test correction. And if I dug in a little bit deeper, I found that CTNS also came up in BioView as being associated with gingivitis, dermatophytocytosis, and influenza. So it seems like it's possible you could create a hypothesis for this gene that it seems to be potentially involved in infectious risk. And obviously, that would be something that would, this is hypothesis generating, not driving conclusions. So this would be something that could be potentially worth exploring further. So next, I went ahead and dropped in the PheWAS code for dental caries and identified a number of different genes that came up at genome-wide significance for dental carie phenotypes, for dental carie phenotype, again across a wide variety of tissue-specific expression levels. And once you have the results, the question is, what can we do from here? One thing that we've created at Vanderbilt is a pipeline where genes like these that are coming up that are of particular interest, where there's robust evidence coming from our EHR records that these are particularly of interest, we have created a pipeline that goes basically straight into zebrafish, where we can immediately start exploring functional studies to look for effects of these genes. But the other thing that we have been able to do is repeat this prediction algorithm exploration in other large DNA databanks. So for example, in the UK Biobank, we performed exactly the same expression prediction models and can do exactly the same association testing. However, UK Biobank doesn't have quite as much dental data in it, but I thought it was pretty interesting, at least at midnight last night, I thought it was pretty interesting, that the gene INVS, which came up as genome-wide significant in BioView for this array of phenotypes, also came up in the UK Biobank, where, again, we don't necessarily have as accurate of measures of dental phenotypes, but came up for a bunch of traits that we know to be significant comorbidities of dental traits, and specifically dental caries. Similarly, another gene that came up as genome-wide significant for dental caries in BioView also came up with obesity-related phenotypes in the UK Biobank. And a third one also came up with not just issues relating to obesity in the UK Biobank, but also impedance measures and Lipitor, suggesting that maybe this is part of a slightly different phenotypic outcome spectrum that would include some cardiovascular measures. So, in summary, existing big data resources can provide exciting opportunities to generate hypotheses for genetic epidemiology. I wanted to really take the time to emphasize today how ancestral heterogeneity is powerful when used correctly in genomic analysis. we're facing a scenario where if you're going to identify low effect size variants, you're going to need extremely large sample sizes to be able to find genetic effects for complex traits. But the caveat there is that we're getting better and better at aggregating large genetic repositories through EHRs and DNA biobanks, which are now publicly available. And what I did last night playing with this portal, any one of you could have done last night as well. It's not proprietary in any way. And in addition to that, functionally orienting analyses can improve interpretation and power so that we can actually know what the results that we find mean. I think one of the key issues, and something I hope we discuss in the next session of discussion time that we have, is how we can begin to work on bringing together oral health measures into EHRs in a more effective way so that DNA databanks can be as powered in those scenarios as they are for measures like central adiposity and obesity. And also that integrating additional omics measures as well as environment is going to be something that'll help us power G by E effects as we move down the road. My lab is small today. If any of you know motivated grad students or postdocs, send them my way. It's a lot of work from 3 people, but of course we don't do it alone. So this is the set of individuals that contributed to the transethnic meta-analysis, and then this is our local group at Vanderbilt that has performed most of the PheWAS analysis within BioView and is responsible for BioView maintenance data aggregation and gene-based prediction algorithms within BioView. And so with that, oh, and then of course, all of the different institutions that have contributed data to GTEx. I'll take your questions or we'll move to discussion.